Hello. I am back, as you can see. Today I will talk about Venom, and I will talk about very fruitful collaborations that we have with the Technical University of Denmark in Copenhagen. So it has been a long-term collaboration that we have with Dr. Lausten's lab in Copenhagen, and we have been publishing many, many papers. The last one just came out actually 2 weeks ago in Nature Communication, and we also have a couple of application reports that you can find on our website. This lab is mainly interested in ion channels and venom, so venom production, and how they can find venoms for venomous species. So they are working on Scorpio, spiders, but today I will especially talk about snakes. So why are we talking about snake bites? Snake bite is recognized as a neglected disease by the World Health Organization, and actually there is every year 5.4 million people that are bitten by snakes. Out of this 5.4 million, we have around 2.7 million people that are envenomed by the snake bites. And within these numbers, there is between 81,000 to 138,000 deaths per year, but also up to 400,000 permanent disabilities like amputation. So this is very present actually mainly in the sub-Saharan in Africa, but there is also there is a lot of death from the snake bites in Asia and South Asia, mainly. Obviously, those snake bites happen in those social, economical disadvantaged communities that they don't have access to good treatments and fast treatments. So there is a huge need to develop those anti-venom and for the production of this anti-venom that can be more efficient. So actually, anti-venom, they have been around for many, many years. And today, we have access to those venom that are developed from this animal immunization. So this is what we call the traditional anti-venom production. They are mainly made from horses. So those animals, we inject the venom from the different snakes, and then we just collect the plasma of those animals, and then we just purify to get the antibodies. And we put those antibodies into a solution that will make this antivenom reformulation. So the problem with those venoms is just they create a lot of side effects. They require huge amounts of antivenom when we have to inject them to people, but also they are quite costly. So we have to use big animals, and there is a huge variations batch to batch due to the fact that animals are quite different between themselves. So the efficiency and the specificity can be low, but they have lifesavers, but we need to find a better way to produce antivenoms. So it is quite time to modernize this antivenom production, and we are working with this lab, so at DTU, that are producing those DNA recombinant antivenom. So those DNA recombinant antivenom are based on DNA, so recombinant and DNA technology, and this phage display technology. So because we need something that maybe comes from humans to make sure that we can avoid big allergy reactions, which is the case when we use those big animals to create antibodies, because there is a lot of side effects and sometimes anaphylactic shock. Then here we actually, so most of the toxins that are in venoms in different snakes around the world, we know them. They have been characterized. So here we can use this phage display library. So we take genes that are known to code for neutralizing antivenom properties, or even fragments of antibodies. And then we use that and we use them into targets. So we just transfer them into bacteriophages or cell lines. And then we use like a purification cycle that is called panning to increase selectivity and specificity. And after that, we just use sequencing, we just amplify them, and then we just create those recombinant antivenom. Those recombinant antivenom, they are more costly to produce just because of this R&D phase and this development. However, on the long term, they are actually way less expensive to produce for scalability. And the big advantage is that you have a better scalability, but you also have a better affinity to the different antibodies and the different toxins. And you are reducing the use of animals. That's what we're all trying to do in research today. So snake venoms are quite complicated. So every venom from different snakes, they are contain dozens of different toxins that we can classify in different populations. Oops, sorry. The main known, the one that are the most present into the snake venoms, especially, so today I will talk mainly about this sub-Saharan snake that is called the cobra, and that we probably have all heard about. But this is like this phospholipase A. So phospholipase A2Es, but also I will mainly get interested into this big group of three-finger toxins. So these three-finger toxins, they are this postsynaptically acting three-finger toxins that we call alpha neurotoxins. So they are mainly acting on nicotinic acetylcholine receptors. So those different toxins that we can find in venoms, in snake venoms, they have different kind of acting, acting mode, so mode of action, sorry. And then today I will talk mainly about this mode of action, which is create this paralysis of the muscles by acting on the nicotinic acetylcholine receptors, and that can lead to respiratory arrest and death. So as I mentioned, today I will talk mainly about a human antibodies that has been developed at DTU. So it has been made by this phage display technology. And this is an antibody that will block these alpha neurotoxins that we can find in the cobra venom. This is also called alpha cobra toxin. And so you probably all know the alpha-1 nicotinic acetylcholine receptors, which is a ligand-gated ion channel. And here we are actually using a human-derived cell line that we have in our lab, which is a stable cell line that is endogenously express the alpha-1 nicotinic receptor. So here, this is the alpha neurotoxins, which the one is called alpha cobra toxin. And the mode of action is that they are entering in competition with acetylcholine, and then they just bind to these nicotinic receptors. And then acetylcholine, they cannot bind to these receptors, which creates this neuromuscular disability and paralysis. So now this recombinant human antibody will actually neutralize these toxins right here, and it will block the access of those toxins to the receptor, which then obviously will neutralize the effect of these alpha neurotoxins on the receptor. So the first thing that we have made at Sophion, actually, so we are very close by. We are in Copenhagen, so the DTU people that actually receive a research grant from Sophion, they came to Sophion with their own venom, with their own antibodies that they create in their own lab. And the first goal was actually to understand what is the dose that we need from this antibody to neutralize the alpha cobra toxin. So the first thing that we made is just like we evaluated what is the EC80 of the acetylcholine to activate 80% of the nicotinic receptors on those cells. So they give us like this is 17 micromolar. And then the second thing that we did is like we use the QPatch2 to evaluate what is the IC80 of this alpha cobra toxin. So what dose do we need to inhibit 80% of the nicotinic receptors when we use this alpha cobra toxin on this machine? And of course, now we need to understand what is the dose of the antibody that will neutralize and prevent the action of this alpha cobra toxin on the nicotinic receptors. So to do that, we use different doses. So we made this concentration response from the QPatch2. And you can see, so in orange, there is always the acetylcholine effect on the nicotinic receptors that will, of course, give a big response, a big current on the nicotinic receptor. And then when we use the toxin at 4 nanomolar, we almost block the entire currents. And we use also this antibody at a very low dose, which is 0.62 nanomolar, that has no effect and cannot neutralize the effect of the toxin. However, when we use the antibody at a dose of 25 nanomolar, then we can completely prevent the effect of the alpha cobra toxin on the nicotinic receptors. So they came with many, many different antibodies that they have created with this phage display technology in our lab. And so we tried them, and the building of this concentration response with these different antibodies, it allowed us to make kind of a ranking of which antibody is the more efficient against this alpha cobra toxin. As I mentioned before, snake venoms are very complex, so they contain many, many, many different toxins. So it's not only one toxin per venom. Some of them actually contain more than 300 toxins per venom, which makes things very complicated. So it also means that we are in the need of producing antibodies that will have this cross-reactivity against different toxins, because you never know which snake beat you when you are in the jungle or in the deserts. So you need to be able to find an antibody and a treatment that will have an action on many, many different toxins. And so we just in need of this high affinity, but this cross-reactive different antibodies. Right now, today, what we have is this animal antivenom that are very specific to one toxin. So each snake have different toxins, and as you can see, right now we have one antibody for one toxin, which can be sometimes efficient, but sometimes, unfortunately, it's not enough. So the whole goal is to actually find these antibodies that will interact with many, many different toxins. And to do that, and to test the cross-reactivity of those different antibodies that have been produced at DTU, we use our higher throughput, the Cube 384, to test the cross-reactivity of those different antibodies against 6 different alpha neurotoxins coming from different snakes. So it's not only cobra, but it's also the mamba and different snakes from this region of the some sub-Sahara. Here you can see it's a heat map from a plate. So it's 384 cells that you can test at the same time. Here is the 6 different neurotoxins, and here we tested 15 different antibodies and a control right here. So this is a heat map that shows the capability of the antibodies to neutralize the effect of the neurotoxin. So when it's red, it means that it worked. So we can rescue the nicotinic, we can prevent and we can keep these nicotinic answers from the cells. As you can see, there are some antibodies that are very specific to only one toxin. But there are other antibodies with the purple arrow that can have a neutralizing effect on many different toxins from different snakes. So this is the whole goal. So finally, we tested, we don't have an animal facility, so at DTU, they tested this antibody that they have developed in vivo. So they injected the mice with a cocktail of this anti-venom antibody, but plus the toxin. And you can see this is, so in black, it's not really visible, but all the control mice that have received only alpha cobra toxin, they died within 30 minutes. so very fast. However, with the different ratio, molar ratio of the alpha-cobra toxin antibody, after 50 hours, we could make them survive. So, of course, we also, so the next project that just been published 2 weeks ago in Nature Communication, it actually shows a different project. So after doing that and using only one antibody against this alpha-cobra toxin, we realized that taking those monoclonal antibodies and making them into an oligoclonal cocktail with many different antibodies, it might be very efficient. So they develop those different antibodies. They make sure that those antibodies, they have affinities against different toxins, and then they make them into this cocktail that they also decided to use. So for this time, they actually used not these human antibodies, but they use these nanobodies. So the nanobodies are those very small parts of a normal antibody. They are large molecules, but they are very thermostable because we know that those human antibodies, that's this immunoglobulin, they are very efficient. They are more physiological because they are made from humans. But the thing is that they are not very stable ex vivo and at a higher temperature. So they also try different kind of technology to produce nanobodies that are way more stable and with a higher affinity. So this is how they made these nanobodies by using, so they actually use blood from llama and alpaca, but that were immunized from a different study by a snake, by the cobra and the mamba. But this time, they use a different snake. So they use the coral snake, which come from the Americas and Mexico, so which is completely different, and to see if we could use this cocktail of antibodies against a different snake venom. And as you can see here, so we tested the same things on electrophysiology, and here we were able to see that this cocktail of antibody, the pink one, was quite efficient. And here, this is the in vivo neutralization of this cocktail of antibodies. This is the green one that make the mice survived. So in this study, they actually injected the venom directly, so completely alone, so without this, not in combination with the antibody. And right after the venom, they injected subcutaneously this cocktail of antivenom, and they could see that the mice survived. So in summary, there is a new whole generation of antibodies that are made against the snake bites that are really necessary for the world right now. And there is a high possibility to use this DNA recombinant technology to produce human immunoglobulin and antibodies that will neutralize the venom. So we used automated patch clamp and high throughput to characterize the different cross-reactivity, but also those of this antivenom and against the different toxins. And we were able to go through the animal model, so which is the mice, to neutralize the effects of the snake venoms, even with multiple toxins inside. So finally, of course, I would like to acknowledge the work of Melissa Bernard-Valle that received a research grant from Sophion to do all this work at Sophion Bioscience by using our machine. We have been Kim, who is a senior research scientist at Sophion. He's the mentor, and he has been working on this project for the past 6 years. So he's really the venom guy in our lab. And of course, I would like to thank DTU and IonTAS that make that it's just like a spin-off of a company of DTU people. So again, you can scan this QR code if you would like to apply for grants. And I would like to thank you for your attention.
Thank you very much. The paper is now open for discussion. Yeah, I have a question. Yeah, thank you for wonderful talk. I wonder if you could make antibody against a common polypeptide lesion which highly conserved among different snake species. Is it possible to develop such antibodies?
Yeah, so I think the phage display technology, it's the whole goal, right? So it will take, because we know most of those toxins, so we will create antibodies, but unfortunately, there is a too big diversity between the different snakes coming from different regions to have one common one. But I think this is the whole goal, to have a cocktail of antibodies that has huge affinities for many different toxins. But, yeah.
So I think that a cocktail will be a very efficient way, but maybe you can design cocktails in the different combinations for different regions on the Earth. Yeah.
Of course.
Yeah. Comment or question, please.
Thank you for a nice talk. This is not the question, this comment. I'd like to ask that you have shown that the BMO has a very toxic for the many people, but I think that the effect is not only the direct effect, but also it has a mediated by immune system, anaphylaxis shock or something like that. So have you tried to think that the possibility to use the immune cell in your system?
Oh, you mean to use immune cells to make antibodies?
Yeah.
That's a great question, but again, you can see. I mean, I think it's one of the possibilities. I think it's just like snake venoms have different mode of actions, so I think we just tried to target the highest population of toxins that we can find in different snake venoms. But of course, I agree. I think that the possibilities are high. Yeah. Thank you.
Hi, please.
Thank you for beautiful presentation. And lethality was dramatically decreased by injecting antibody in vivo. And was there any inconvenience in the animal?
So for the cocktail and for the data that I showed, actually there was absolutely no traces of respiratory disability or any toxicity of those cocktails. But some of the other cocktails that we made, yes. So some of we checked the mice during 6 hours after injection, and some of them, they also showed sign of respiratory or allergic reactions. So it doesn't work all the time. Yeah.
Any other
and you have to be very fast of injecting those antibodies after receiving the venom. So it means that you have very, the window, the time window is quite short.
Okay. Okay. If no, let's close the session, and let me please thank all the speakers, and I also appreciate hot active discussion. Thank you very much.